From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. This episode was recorded on September 11th, 2023. Hello, Micronauts, and welcome to our Quality Quorum today. Today is the 7th episode of Matters Microbial. Thank you all for listening and or watching. First, another denizen of my Wunderkammer. Now, a former Micronaut student of mine, Anne Zeller, had this made for me. It's a gooseneck flask of the style that Louis Pasteur used when he put paid to the idea of spontaneous generation. Now, this was a big deal in the 19th century because the germ theory was relatively new. So what Pasteur did is kept a small amount of nutrient media here that was sterile and was able to show that even though air could travel, it didn't instantly become contaminated here because some thought, believe it or not, that the media could just naturally have life happen. And Pasteur wanted everyone to know it was coming in. So what he could do is bend that so that some of the media would hit the dust that was collecting in the gooseneck, and then that would actually lead to the contamination. It's something I've been after for I can't tell you how many years, and I'm very, very grateful. I will link to that story in the show notes. Now, for today's inspirational lapel pins, the first is by Liza Vanderart, friend of the show, and it shows the fascinating organism Colobacter crescentus. Now, that's well adapted to low nutrient conditions, oligotrophy, that we've discussed several times. Liza, when she sells such pins, includes an informational card that is a lot of fun to read and a good job of science communication. The other pin that I'm wearing, sometimes problematic, but I consider a lot of jargon to be needlessly exclusionary of people. I'm the sort of person that wants to be able to explain what I do to everyone. So that's part of what I want to believe. The second week of my wonderful microbiology class, Bio 350, here at the University of Puget Sound, my Micronauts did a certain degree of lux art where they painted onto nutrient media with photobacterium, and you can see their creativity here. And I will link to a video of this in the show notes as well. We also started to explore microbes in their reusable water bottles. Now, this bothers some people. They kind of get upset with it. And I kind of remind folks that we exist within a microbial cloud at all times, as this drawing from a former student of mine illustrates very, very well. Now, the trouble is, and I've alluded to this in earlier episodes of this podcast, is that many of us sort of bought into this idea that I call devil microbes, as you can see from the illustration, where all microbes were bad. That's not true. With the advent of all microbiome studies, other people went as far as they could in the other direction and started telling us how all microbes were good, which I call angel microbes. Also incorrect. The fact is, for the vast majority of the microbial world, we're not even important at all. What I call meh microbes. It's important to remember that when we look at questions like this. In fact, another way of looking at it is thus. The number of microbes that can cause disease, we can call them pathogens if you like, it's vanishingly small. Think of them like juvenile delinquents of the microbial world. Very low in number, but they get all the press. This artwork has been done by Maggie Middleton, and I have given you all a link to her wonderful Redbubble page where you can get like microbially themed clothing, which is awesome. So, from swabbing in the reusable water bottles, my Micronauts saw a number of things. Here is how all of their plates looked. As you can see in this, lots of different denizens that were growing there on the media. However, when I swabbed from the water bottle refill station or a standard water fountain, I saw lots of microbes there as well. Now, is this bad? I rather doubt it. We're surrounded by microbes at all times, and these are surely adapted to low nutrient concentrations, that oligotrophy we talked about earlier. And in fact, if you look at this particular photograph, I believe we found, and we've seen this before, antibiotic producing members of the water bottle buddies. And I wouldn't be at all surprised since they're kind of duking it out for space. It is true that we exist in a sea of microbes. We only just have to look, and everywhere we do, we see it. And Dr. Elinne Becket of California State University, San Marcos, is joining us today to describe some of the work she and her wonderful undergraduate students are doing, and to explain an extremely popular hashtag on social media, Blue Soup. Welcome to our Quality Quorum, Elinne.
So great to be here. Thank you so much for the invite.
Oh, it's such a pleasure to have you here. And you know what else? I believe this is the first time I've seen your face.
We met at ASM, remember?
Well, the first time we did, we were masked.
Yeah. Oh, good point. Yeah, 100 point.
Yeah.
Okay, that's what you mean. Okay, this is the first time you've seen this half of my face. Gotcha. Okay. Yes, fair point.
So I think a really good place to start is to explain a little bit about the Cal State system and the kinds of students that you have in your lab.
Yeah, so the Cal State system, we're 23 campuses across California. I think it's the largest university network in the country. And we're what's called like regional universities. And so we mostly, with some exceptions across some of the campuses, but mostly serve regional student populations. And so it's really cool how the vast majority of our students are from the area. And most of them, I think over like about 70 to 80% of them even stay in the area in the local workforce. And so especially in San Diego, it's such a huge biotech industry and biomedical industry, just tech industry in general. It's such an opportunity for our students to go into the local industry over here. And because of all these factors together, and because my colleagues are so incredibly dedicated to our students, we are this year the #1 in the country in social mobility. And so, yeah, if I talk about rankings, I'm like, forget all those other rankings. That's the ranking that I want. We help our students improve their lives as much. Actually, they help themselves. We help guide them, help themselves improve their lives as much as possible. And so it's just, I'm just so grateful to be part of this particular university and to serve our students that we have here. It's been awesome.
So before we begin with the story of Blue Soup, which dying's a strong word, which I'm very interested to learn more about. Can you tell me a little bit about, and I have a link to your laboratory website in the show notes, a little bit about the general thrust of research in your lab with undergrads?
Yeah, so I've had quite a twisty past experience in research. I've gone from drug testing in sports to bacterial genetics to cancer epigenomics to being in industry to developing assays. And but early in my career, when I in my first year of grad school, I learned about how specific bacteria in response to exposure to stressors such as antibiotics, they actually start sharing genes with each other. It triggers it. And so for me, I'm just here are these little individual non-sentient cells that in response to a threat, they're sharing information that can make them more fit in that particular population. And also I'm just interested in to whom do they share it? Which ones are excluded in that, and how far does this go and what are the triggers? And so I've been obsessed with that for so many years. And then finally, I'm able to have a lab that really focuses on how external stressors cause not only microbial populations to evolve based off of who can tolerate those stressors or they grow better during those environments, and also then now how these these pieces of DNA are shared in the genes that are carried on them within the population. And so that's really been our focus in our lab. And also since I have industry experience in developing assays, and I predominantly train undergrads, they do the entire pipeline start to finish. So they go from collecting the samples, mostly in the coast. So it's really great to have beach days as part of our as our office some days. And then they go through the whole like the DNA extraction, the sequencing library prep. We have a small sequencer in our lab, the Illumina iSeq, that we can do like some quick test runs. And if we need deeper sequencing, we'll send that out, and then do all the computational analyses. And so it's just so cool that like when I didn't even do a PCR, in my undergrad, so I was a biochemist, to be fair, but still, and but for them to be able to, for me to be able to do with them, like these full start to finish next generation sequencing pipelines, to be able to answer some of these questions, and how basically the main central question is, how is all the crud that we're dumping into the coast affecting our coastal ecosystems, because microbes make the vast majority of biomass in our ecosystems and then determine the overall health of those ecosystems. And so so that's really kind of the crux of what we focus on.
Are you familiar with Julian Davies at UBC?
Of course, one of my one of the folks that have inspired this line of research, for sure.
Well, he and I talked many years ago, and this idea that he has that low levels of antibiotics have effects that you're not expecting is just fascinating to me. And he actually told me once that he wanted an ohm of his own, because he was tired about everyone having ohms.
The everything ohm.
Right. Yeah. So I gave him one.
Okay.
And I just am telling you, if you've ever seen this in his work, I'm responsible in a way. This is shining by reflected glory. Parvus is Latin for small. So parvum means the small molecules that are leading to these kinds of effects. And it certainly has influenced the way that I look at the way the antibiotics work. And honestly, I'm so glad that you do research in this area and teach about it, because we don't teach about antibiotic resistance enough, do we?
Yeah, and if we do, it's always in a anthropocentric, no jargon. It's in a human-centric focus. You know what I mean? And and so it's like, it's so much more than that. And also the fact that people don't realize that, even though the misuse of antibiotics has spread the issue of antibiotic resistance, that it is an ancient thing. It's the result of millions of years of chemical warfare between microbes. And as you mentioned earlier in your with the plate that you discovered. Yeah. And so with the antibiotic production comes the necessary to have defense systems and antibiotic resistance. And so there's so much antibiotic resistance that is naturally in the environment that has nothing to do with clinical stuff. And so, but it's all about how these antibiotics, how these microbes interact with each other, and how these communities are formed. And so it's absolutely fascinating, because really, it's beyond the comprehension of humans, like how dynamic these factors are.
It's so funny to me, because the first tattoo I ever got, I'm looking at it now, Ovite Pawele Domini, all hail the small masters, because that's what we're dealing with, aren't we?
Yep.
Well, that's a wonderful thing. And I just wanted to ask those questions for myself, though I do think it's super important for listeners and watchers to know about this work. And again, your website goes into some wonderful details on it. And any pictures that you'd like me to share, or papers, just send them my way, and I'll put links to them in the show notes.
Great.
I would love for you now to tell the story that must be told.
Oh, the fact that as my PI says about so my PI is Jeffrey Miller, and so one of the things he's known most for is Miller units, which is just an arbitrary measurement for beta-galactosidase assays. And he says, he's like, I am most known for my least important contribution to science. And we don't yet know how little this contribution to science is, but at the moment, its contribution is joy and curiosity. And so let's not underplay that, for sure. But still, what's been so great about this story is just especially after the last 3 years of what we've gone through, bringing people together through joy and curiosity and reminding us why we love science and why we love microbiology is just like, that has been the absolute best thing about this. And so it happens just from me being me, which is I'm not great at cleaning out my fridge, and so we all have our weaknesses and our vices, and that is one of those for me. Once it's gone past a certain point, I'm like, I'll get to it when I'm in the mood for that, for sure. I know. So my amazing mom, who expresses love through food, among many ways. So she makes me this beef soup every year for my birthday, because my birthday's in January. It's freaking cold. The soup warms me both emotionally and physically, and so I look forward to it every year. She's like, "I'm gonna mix you something else." I'm like, no, same soup every single time. And so it's like, it takes like 5, 6 hours to do the whole thing. And so she makes me this vat of it like every year. And I had it like 3 meals a day for like a week plus, right? And I really need those, like, I've learned that today, I really need those like soup cube, like ice trays, so I can actually, so this cannot happen again. But then in the pot in the back of the fridge, like there was like maybe this much left, right? Of like, from like the vat. And I'm like, at this point, I was like, okay, I'm past this, and I'll get to it when I get to it. Well, I didn't get to it. My fiance got to it 4 weeks later, and was like, I'm just going to clean the fridge and throw everything out. And then he like gets the soup, and he's like, is it supposed to be blue? Like, and I'm thinking, maybe a hue or something. And I'm just like, what are you talking about? He's like, no, seriously, look. And I look, and it was the brightest azul I have ever seen. And like, it was just like this awesome, and it was gorgeous color too. If like, aside from the fact that that it means that something is growing in there. And so I'm like, what? Even after 15 years of microbiology, like, I'm just like, this is too much for me. And so we poured it into like a double bag, and tied it up and threw it in the trash. And so then, of course, I like immediately tweet my reaction to it. And I should have known better than to tweet something like that to thousands of microbiologists, because I should have known they would have not let me get away with this. And they're like, take a picture, take a picture, take a picture. And I'm just like, y'all, I'm not going back into the trash. Like, are you kidding me? Right. And so, of course, they nagged me enough. So I went back in the trash and I opened it up. I took a picture of it in the garbage bag. And even then, in the dark garage, because it was like 10:30 at night, like that bright blue was there. And then that's when everything erupted. Everyone's like, take a sample, take a sample. I'm like, okay, y'all are now across this. So they spent an entire day trying to convince me to take a sample. And then finally it was, a lot of different people goaded me. And especially I know Michael Boehm knows how to push my buttons. And I was like, almost got me. But then Pedro Leao and I hope that's how he pronounces his last name, and Sebastian Kochoba, they went into a back and forth about he's like they're like, gross is temporary, glory is forever. And went on. And so they finally convinced me to take a swab of it and go back in the trash again. Thankfully, it's cold. It was cold enough in California that it was refrigerator temperature for the previous 24 hours, and so it was unchanged in the day that it had come. So then Sebastian's like, you need to take at least this many samples for this many analyses, and I'm like, oh, I gotta go back in there.
Yes, you do.
So I went back
yes, you do.
Back a fourth time. I got all up on my arm and stuff, because by then it's on the bag and stuff and everything. And I didn't realize it actually had extra swabs for COVID tests at home. And I should have used those. Instead, I just grabbed some non-sterile Q-tips, or as Sebastian says, the domestic Q-tip and everything, as opposed to the Q-tip found in the wild. And so
small batch, artisan grown, right.
Lovingly crafted. Anyway, but so I got those, and I'm like, I'm never forgiving you for this, Sebastian. So because our lab is equipped with start to finish everything, I was able to quickly extract the DNA, and I sent one bullet of it with one swab and a stab to Sebastian, and then I kept a second copy of it here in case it didn't travel well, because he's the one who's much more equipped for all the microbe streaking and testing stuff on that, very similar to you. And I do much more of the genomics and the molecular side of things, and that's more what our lab is equipped for. And so, but I also like swabbed like one of the swabs on a plate, which was made with LB plus beef bouillon. And so I just grabbed a beef bouillon cube in it, and I'm like, that's what's in the soup. So maybe that'll like help it grow.
Oh
And
my.
just to keep everything alive, just in case that
Sure.
it didn't travel well. Uh, and so then, yeah, like I made like sequencing libraries out of it, and I sent it off to Sequoast because they were like, "Gimme, like, let me sequence it." And then before I could even look at the data, like their bioinformatics team was like, "We're on it," and went through and dove down, and they found that most likely, based off of the metagenomic analysis, that it is Serratia quinivorans that is producing indigoidine. And which was a total surprise because everyone thought that it was like a pseudomonad, like one of those, because previous work from folks in the Jonathan Eisen lab had isolated blue microbes and found that it was like a pseudomonad producing that blue pigment. And so it was a surprise. And what was even cooler was that Sebastian and we streaked stuff out and we threw our plates in the fridge, and because life got in the way, we forgot about it. And then like 3-4 weeks later, only the ones in the fridge turned blue, but the ones that were on the benchtop, like just even at room temp, like were like didn't turn blue at all. And so we're really, really interested in like, whoa, like this is definitely something that's being produced in response to the cold temperature. And it's been known that some Serratia species, like at like 37, they won't turn like a particular pigment, but at room temp they will. But going actually this low is interesting. And so a few things I saw is that it might be a cryoprotectant, so protecting the microbe against cold temperatures. And then also it could serve like some antimicrobial properties as well. And so we definitely want to like dive into that. But yeah, so that's kind of like where all that went. And I realize I'm rambling for a while now between questions, but like it's just so cool. I've got this multi, and not only that, but everyone was like, have you thought about this? Have you looked at this? What about this? This is my hypothesis for that. And so it's just like, it is the ultimate team science and the ultimate open science. And then the pinned tweet on my Twitter profile, I will call it Twitter forever, I do not care what he says, is the entire like live lab notebook of what we've done so that people can see the entire step, both what I did and Sebastian did. And so we're really, really both super, super passionate about this being like an open science endeavor, and for all of us to share the joy in what the heck is in the back of my fridge? Because let's be real, everyone has asked that question before.
So 2 things really quick, and I do have to use a little bit of jargon. Psychrophilic things, that is to say, organisms or enzymes that like the cold, are fascinating. And I don't know if you know the Ned Ruby story about this.
Remind me, because maybe I don't know the jargon of that particular organism. I'm like, there was this one bug, but anyway.
So bioluminescent bacteria are all with this blue-green color, about 490 nanometers, because that transmits very well under conditions of seawater. And when Ned Ruby was a graduate student, he would have all these things collected off Scripps Pier, and he had them in the cold room because they would still grow. And he saw this one plate that was glowing yellow, which no one had ever seen. So he was so excited, he takes the plate out of the cold room and, he streaks it out, puts it back in the cold, and now he has this plate full of single colonies that are glowing, not fluorescing, glowing with yellow light. Unheard of, unseen. So he runs up to the campus from Scripps carrying one of these plates to show it to someone who knew something about bacterial bioluminescence, and lo and behold, it was back to blue-green.
So it's temperature change, yeah.
So what it was is there was a chromophore that absorbed at 490, re-emitted at the yellow wavelength, but that chromophore was temperature sensitive. So it's easy to take all of this personally when the microbes do things like this to you, right? But to me, it's a wonderful thing. So I just wanted to,
like you said, the vast majority of microbes don't care about us.
Yeah.
Yeah. So yeah, exactly. So definitely, it's exactly along that ilk. So you just gotta be okay with that and realize that you're just a speck, like on the behind of the universe. So.
And what wonderful work because I would have thought there would be so many different things you would have seen. With all due respect to your mother and her culinary excellence, I would have expected so many different things to be there. But are you saying only one type of microbe grew?
No. So there was definitely like 80 some percent were pseudomonads.
Yeah.
Like definitely a chunk of those were specifically pseudomonad fluorescens.
Yes.
And so those were the largest chunk. And so that's when Sebastian streaked stuff out. That's probably a lot of what he got. Because he did see a lot of ones that did fluoresce when he put them on specific media. And he also, the one that actually really we want to look at is there's one that is iridescent. And so we kind of want to check out, like, what the heck is that? It's like almost looks like an oil slick. And so he's got a few strains isolated that I definitely want to throw onto the sequencer to see, like, what the heck is this?
How wonderful.
Yeah.
How wonderful.
It'd be really, really cool. So it's it'd be really nice to just discover, like, maybe we might discover new strains or new genes that haven't been identified yet. So I mean, so much of like the birth of various molecular or microbiology fields came from these, like, what's going in my bread, type of thing. So,
oh, I couldn't agree more. This is such a wonderful thing. And, I've been doing a certain degree of the Tiny Earth program that Joe Handelsman runs. And I've been doing it with my first year students. And when they find something I've been looking at lichen and mosses because I thought they might have ways of excluding other microbes.
ooh.
And we were able to find a lot of things that were making antimicrobial agents. And this first year students find something that's active against all the 7 groups of classes of microbes that are there. And the look on their face is like, and I said, you did this.
Yep.
So your good friend Mark here is trying how to learn how to do genome announcements all on my own to get that published.
Well, I may or may not have been or I'm currently an editor for Microbiology Genome Announcement. So here to help as much as you need.
Well, you must understand that I'm an aged canid, but I every time that you talk about your lab, I want to come work there. So keep that in mind.
You should come visit, hang out. We got we got like a half wet, half dry space and everything with the best chairs. Yeah, that was best money spent ever. Yeah, for sure.
Oh my.
Yeah. But so I do want to really quickly point out that this contamination came post cooking. Like, sorry, that soup was boiled for many, many hours, and my mom's cooking. I know you didn't insinuate that, but I mean, but there was enough people that insinuated, like, things about my mom's cooking. So I guarantee you this was from me going in with a ladle like a few times a day for like a week. So it definitely came from that stuff.
Well, anytime that you leave the tops off plates just in lab, you get all kinds of interesting things that float in on the air. So no disrespect anyone should give towards your honored mother on this. That's anyone who's looked in the back of their fridge has seen unusual things that have nothing to do with cooking.
Oh, yeah. My mom is so pure that despite the fact that over 1 million people have interacted with this story, and yet her response to that is and probably at least a quarter of Croatia at this point, because we're Croatian and the recipe is a Croatian thing. And her response to that is to write me a sonnet called Ode to Blue Soup, have it framed and sent to me. And so
Can we put a picture of that up on this? This is what a wonderful idea. Oh my goodness.
So I mean, like that's how pure, my mom is throughout all of it. So, but it's half purity and half like wanting to make fun of me, because it's like I'm going to point out my daughter's terrible fridge situation, but at the same time, here's some iambic pentameters for you at the same time.
Well, that is a beautiful story. So where's Blue Soup going next?
We're trying to draft a publication on it. And the last thing I want to do is I want to isolate the blue from the media that is being excreted. But My lab fridge may or may not have suddenly died, and so yeah, between like having to shuffle stuff around, I'm trying to like regrow it and isolate the blue so that I can actually get the chemists involved and hopefully
Yes.
like do prep TLC and NMR to see like, is it actually indigoidine, like, or is it maybe some slight different version of it, because if it is something different, they'll be able to pick it up. The biosynthetic pathway from the metagenomic data suggests that it is indigoidine, but it's a homologue of the indigoidine biosynthetic pathway. So you never know, it could be slightly different. So here's hoping, but either way, it's worth publishing. And at the very least, the methods section and the acknowledgement section will be worth the read. So,
You've got to include the sonnet in there. Come on.
oh yeah, as a supplementary table 1.
That's it. Yeah, that's the way it normally is. What a wonderful story. And you got me going on it. So I know we're a little short for time, and that's my fault, Logoria, speaking of jargon. But the whole thing that I wanted to do now is I like to ask, we've covered most of the questions I would normally ask you in other contexts, but this is the one I like to kind of leave speakers with. There goes my lights again. I'm a weeble for this. And what I would like is to ask this question. What one particular event sold you on being a microbiologist?
That's the thing that's really tough. It's a combination of circumstance, because the amazing mentor who took me under his wing is like one of the OG microbiologists,
Yes, he is.
and so that like that definitely. But I think fundamentally, I've always been fascinated with things I can't see. If I can see it, I don't know. I've never been really like jazzed about it, and even microscopy, like never really got, because I'm like, it just assisted seeing, you know what I mean? Not just obviously like that stuff is complicated and amazing and phenomenal, but I mean, like if there's nothing that will let me see it, like, and I can just measure it and then imagine it, like for me, that is magic. And that's what I've always been. I've been fascinated with things that like that can't just be looked at. And so, and especially something as complex as microbial communities and how they interact in this way, like you can only like see the shadows of the effects, and actually like, and so like for me, like that's what captivates me as part of it. And so it's the part of that, and also partly like the microbiology and specifically the microbial ecology community is just phenomenal. Like how focused on inclusion and open science, and like it's just, I've just always felt at home here, and like I'm with my people, and that everyone like everyone goes to bat for my students, like even though like they're not here, you know what I mean? And so it's just like, and likewise, and so it's just, it's a combination of all those three things that are just like, it makes me want to get up in the morning every single day. So it's not like a singular event in any way. It's really, it's really just like what brings joy to my life.
There was a saying when I was a student back when dinosaurs ruled the earth, is that biochemists drop a bomb on a cell and they look at the pieces. That's not really true, but that's what people used to say. Geneticists, on the other hand, look at life as a black box. They see what goes in and what comes out. And I think what you're touching on is the puzzle that we all wonder what's going on in that black box.
Yep.
And so that's
Schrodinger's genes.
And that's what makes me a geneticist instead of a biochemist. But no disrespect to biochemists at all.
No, that stuff is hard. I was a biochemistry major when I was an undergrad, and like, I'm like, y'all with your kinetics and stuff, I'm just like, props, but too much for me.
So that's a wonderful story. And what I particularly like you doing today is talking about the joy that you found. You've heard of the joy of cooking. Well, now this is the joy of microbiology. And it's such a really good thing for folks to think about, because if you're outside the field, you don't understand it. But this is how artists feel. This is how writers feel. This is how people who are crafters feel about some of the things that they make themselves, and how fortunate we are to have jobs like that.
Yeah, absolutely. I mean, it's like, it's the amount of fortune and luck that I have to be able to to do what I do and work with the students that I do and the colleagues that I do is just like so unbelievably serendipitous, and I'm so grateful for it.
You can see the microbial tears coursing down my face at hearing your positive experiences. It has been such a delight chatting with you, the very famous Dr. Becket.
Not famous, but
Yes, you are. You're a very important microbiologist, and it's just been a delight for us to spend the time chatting together.
Thank you so much.
So I hope that we can talk again sometime.
Definitely.
And get updates on all of this.
I'd love it. That'd be great.
Okay. Thank you very much and have a wonderful day.
Thanks. You too.
This has been Matters Microbial, a weekly podcast about the wonders of the microbial world and the people who study it. You can send questions, suggestions, and comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with wonderful links, I should add, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Becket can be found in the Biological Sciences Division of Cal State San Marcos. Many, many thanks to Julia Russo for superb editing and Reber Clark for some quirky music indeed. I hope that you've all enjoyed being part of our quality quorum for today. See you next week on Matters Microbial.